I'm going to talk about GBA2, which is the cytoplasmic glucocerebrosidase. So that's distinct from the GBA1, which is the lysosomal acid glucocerebrosidase, which is implicated in Gaucher disease and also in some aggressive forms of Parkinson's. So there is an increase in emphasis on the lysosome as an organelle which is important in neurodegeneration. And I'm going to be talking about the potential role of cytoplasmic glucosylceramide in activating Niemann-Pick type C lysosomes. So this is a glycolipid talk. Glucosylceramide is, of course, separate from what we get with most of the glycerophospholipids, membrane-forming lipids. We have glucosylceramide, which has got a slightly different structure, got a sphingosine backbone. And even in a yeast, which have slightly different sphingolipids, glucosylceramide is often present, and it's the precursor for a higher glycolipids in mammals, such as gangliosides, etc. Glycosylceramide is also present in mammals and bacteria as well, some bacteria, and it's got a more specific distribution. So my major point is that glucosylceramide is particularly widely distributed throughout eukaryotic system, and not just widely distributed throughout the cytoplasm as well as the cell surface. And one of the things that is particularly important is that as opposed to knocking out higher glycolipids, knocking out glucosylceramide leads to embryonic lethality in mice. And even in yeast, you get clear phenotypes with it being associated with growing under alkaline conditions. We think that glucosylceramide is particularly important. When it accumulates in the lysosome, it causes disease, just like other glycolipids do as well. And it's not very clear why this occurs. Of course, there's some talk about lipid raft accumulation inside the lumen of the lysosome causing disease. We'd like to highlight the fact that cytoplasmic rafts could also occur. In Niemann-Pick type C, we have slightly different. MPC1 and MPC2 are cholesterol transporters or putative cholesterol transporters, and glucosylceramide accumulates in the lysosome as well in Niemann-Pick type C. This is a childhood dementia disease, childhood Alzheimer's, with A-beta and tau accumulating, which is reportedly reduced by miglustat treatment. So miglustat has got FDA and EMEA approval for the treatment of lysosomal storage diseases, an inhibitor, or designed to be an inhibitor of GCS, glucosylceramide synthase, inhibiting the synthesis of that, leading to glycolipid storage reduction by inhibiting the synthesis of glycolipids in the first place. But there is some discussion about to what extent it's a mixed inhibitor. The IC50 is a little bit high on the 40 micromolar stage for miglustat. When you look in the brains of the mouse models, you do often get increases in brain glucosylceramide, and which is not consistent with target engagement for the synthase inhibition. And of course, the other major issue is that miglustat is a very good inhibitor of GBA2, the cytoplasmic enzyme. Really something that is unique among glycolipids, at least. Glucosylceramide is synthesized in the cytoplasm, is rapidly transported in the cytoplasm by transfer proteins, GLTP, and it's rapidly broken down in the cytoplasm by GBA2. This pool is under-recognized. We can't actually measure it directly, but we can modulate it. We can increase it by adding GBA2 inhibitors. We can also do it by adding brofeldin A, which acts as a different target, COP1, to form the ER Golgi, which has a very high level of glucosylceramide synthesis. We can increase the levels of cytoplasmic glucosylceramide. There are, in fact, a lot of targets which are present for cytoplasmic glucosylceramide, which we don't know about. So there are translocators in the post-Golgi and pre-Golgi. We don't know what these translocators are. They are putative drug targets for the future. and even GLTP, no inhibitors of that are known, but miglustat could well be inhibiting that for all we know. Anyway, this is an early story, but we do have some knowledge of what cytoplasmic glucosylceramide might be doing in cells. At least in skin cells, it gives an extra kick for the ATPase for the correct sorting of tyrosinase. But more recently, these are glycolipid negative cells which have a lower 50% of the VATPase activity. But actually, one of the things that we found recently is that actually this is quite a general phenomenon. Endolysosomal pH is increased under CRISPR knockout of GCS, glucosylceramide synthase, pharmacological inhibition, will increase the endolysosomal pH in a variety of cell types. Important. So the question is, can we do it the other way? Can we actually balance up the storage in Niemann-Pick type C fibroblasts by inhibiting GBA2? And can we see increases in glucosylceramide? And indeed we can. In fact, using this rather more potent AMP-DNJ inhibitor, which is related to miglustat, we can see about 4- or 5-fold increases in total glucosylceramide levels, perhaps consistent with what we get changes in non-vesicular transport of glucosylceramide. MPC fibroblasts do seem to be, altering the way they handle glucosylceramide, because you notice that in the control fibroblasts we're not getting anything like the increases in glucosylceramide. I previously showed you when we knock out glucosylceramide synthase, we get increases in endolysosomal pH. So if we increase the levels of glucosylceramide in the cytoplasm, do we decrease the pH? And indeed we do. This CRISPR knockout shows decreased endolysosomal pH of GBA2. So we and others have already found that in Gaucher disease we get a defective acidification of the Gaucher lysosomes, and there's a reported increase in GBA2. Indeed, knocking out GBA2 is disease-modifying in Gaucher disease mouse model and MPC mouse model, and more recently in ALS as well. So GBA2 is of increasing interest for neurodegenerative disease, and of course is reported to be increased, perhaps as a compensatory mechanism for defects in GBA1. Gaucher disease, we've got GBA2 overexpression. MPC reports of increased GBA2 expression and storage of glucosylceramide. So does that lead to defective acidification? And indeed it does. Using a general probe, we can see increases in endolysosomal pH in various MPC cell culture models. And this has been reproduced recently by using a DNA nanoprobe, where it seems to be that a subsection of the MPC endolysosomal system is not correctly acidifying. We're picking this up using our general probe. So the question is, well, what happens? Can we rescue this deficit? And indeed we can, either adding Brefeldin A to increase glucosylceramide levels indirectly, or by adding AMP-DNJ, and more recently we've been trying to do miglustat as well, and that seems to work too. A decreased pH, even in control fibroblasts. So these data sets are really quite similar, and we think that this is consistent with glucosylceramide, cytoplasmic glucosylceramide, activating these MPC lysosomes. We also get changes in Bodipi endocytosis using a glycolipid-dependent endocytosis. Lactosylceramide is trafficked to the Golgi apparatus in normal cells, and this is disrupted in MPC fibroblasts, and this is also rescued on AMP-DNJ treatment. Lysosomal acidity, VATPs being the major candidate for helping this, and indeed we do get increased expression of VATPase on treatment with AMP-DNJ, and we get messenger RNA effects as well, increased expression of the VA subunit. So one possibility is that glucosylceramide binds to the VATPase. We don't know. It could be a raft mechanism, but it could be direct binding too, and we do find a binding site for this. There's been a cryo-EM published recently of VATPAs, but unfortunately it was in baker's yeast, which is one of those yeasts that don't produce glycolipids. So that was unfortunate. So we've got a model where we think that on the surface of MPC1 endolysosomes, we've got a deficit in glucosylceramide. This is possibly either due to an overexpression of GBA2 as a compensatory mechanism, or changes in trafficking of glucosylceramide, which we do also see. That's in the paper that we published in Neurobiology of Disease. More detail on that. I'm not going to go into that right now. This defective glucosylceramide, which we think is the major sphingolipid on the surface of the lysosomes, it certainly could be, it can be mended by inhibiting GBA2. So I'd like to thank Simon, who for most of these experiments, and Meenakshi's doing a poster on our results in other diseases. Thank you very much.
Thank you, Dan. Do we have one or two questions? So you looked at the lipid rafts in the lysosomes. Is there any changes happening to the other membranes, cell membrane, for example?
Yeah, I think you can detect changes by things like atomic force microscopy on the cell surface, so that you do get changes in the cell surface too in these storage diseases. Yeah.
Yeah. Okay. Okay. One more question. Okay.
Sorry, when you say you measure the VATPAs increase, the messenger RNA and protein, do you measure all the subunits or?
No, we only just the A subunit. A lot. I've got a collaborator in the EMBL. A lot of the subunits are glycolipid dependent, or do go up and down with glycolipids. So
you just measure the A.
Yes. Yeah.
Thank you, Dan.
Okay.
Thank you.